In this lecture I'm going to take you through the key points of the cell cycle. So we've got an overview of DNA replication, an overview of the different stages of the cell cycle that we're going to cover, and we're going to get all the way through to the retinoblastoma pathway and how that's controlled by Cdk inhibitors and cyclins. So just as a very, very quick reminder, here's a cell cycle. In G1 phase, you've got the point where cells are actually listening out for growth factors. They're expressing growth factors are binding to the receptors, and that's driving growth somehow. We'll look at the signaling later. In S phase, you've got DNA replication. G2 phase, you've got a lot of checking of your DNA. You've then got a DNA checkpoint there, where all the cells can't enter M phase unless all the DNA is being replicated properly. And then we've got M phase, where you've got prophase, metaphase, anaphase, and telophase, where you get segregation of chromatids and separation into two separate cells. And this is just highlighting what's going on at M phase. So most of the cell cycle is interphase. So you've got G1 in interphase, you've got S phase, which is part of interphase, and then G2, which is interphase. And then you've got your prophase, metaphase, anaphase, and telophase. And you can see at metaphase, you've got your chromosomes on the mitotic spindle. At anaphase, you're starting to separate the two chromatids from each other. One goes one way, one goes the other way. And then you get cytokinesis. So in the cell cycle, this is a classic illustration of the cell cycle. There's your M phase, prophase, metaphase, anaphase, telophase. Cells can go into G0, which is an extended resting phase, and we say cells in that phase are either in quiescence, which is sort of an organized, non-replicative state, or senescence, where the cells are sort of old and damaged and they're non-replicating. During G1 phase, you've got this period here where cells are responding to growth factors and they're responding to anti-growth factors. So TGF-beta is your anti-growth factor, and your growth factors are things like epidermal growth factor, platelet-derived growth factor. This is the first major checkpoint in the cell cycle, which is the R point or restriction point, and this is where the retinoblastoma pathway controls whether the cell is going to progress into S phase or not. This illustrates the cell signaling that goes on growth factor receptor signaling. So EGFR is your epidermal growth factor receptor. That binds to epidermal growth factor. That passes on a signal to RAS, RAF, and then these kinases, MEK and ERK, which we're not really going to go into much detail on. But to cut a long story short, after ERK becomes activated, we get an induction of cyclin D. Now cyclin D is going to be really important later on when we look at cyclins, because it's the first cyclin that goes up in the cell cycle. So EGF binds here, you get induction of cyclin D. Just as a reminder, you've all grown cells in the lab, you've put fetal car serum on those cells. That fetal car serum contains epidermal growth factor, platelet-derived growth factor, and fibroblast growth factor, and that is what's making your cells grow in the lab. This slide illustrates the main checkpoints that goes on within the cell cycle. So we've got the R point here, which is basically whether your cells have got growth factors or not. During S phase, cells will stop the cell cycle if there's DNA damage. At the end of G2, they'll fail to go into M phase if DNA replication has not been completed. And then in M phase, there's a metaphase to anaphase transition, which you'll probably learn about in the proteasome lecture, and that is to do with whether all the mitotic spindles are attached. The cell cycle is controlled by proteins called cyclins, and these are induced at certain stages of the cell cycle and then reduced in their levels when they're no longer needed. There are four classes. The main way to remember them is you've got cyclin D, E, A, and B. So cyclin D is your first one, that's in G1. That cyclin D is induced directly in response to growth factor signaling. After a while, cyclin E goes up. That gets a cell through the R point. Cyclin A is crucial in S phase. Cyclin B is crucial for that metaphase to anaphase transition and mitosis events. The cyclin proteins themselves the levels are low naturally, and then they get induced briefly in certain stages of the cell cycle. So if we look at mitosis and then into G1 phase, in response to growth factor signaling we get a rise in cyclin D, then that is going to be deliberately and targeted and destroyed as cyclin E levels start to rise. So basically cyclin D gets the cell cycle started, we'll find out why shortly. Cyclin E then takes over, then levels of that decrease and cyclin A takes over, and then we've got cyclin B doing the M phase events. Now these rises and falls in protein are due to gene expression, so cyclin D gene gets switched on, we get cyclin D mRNA, cyclin D protein, but then this reduction in levels is targeted destruction. And you want to be thinking about these reduction in levels here when you look over the notes for the proteasome, because these proteins are targeted by the proteasome and deliberately destroyed. And this event here, cyclin B destruction, occurs just in a few minutes. If you're struggling to remember the order of the cyclins, because it seems daft starting at cyclin D and then cyclin E, the way to remember it is that for some people, depending on which organism you work on, maybe the cell cycle starts at S phase, because that's the first time we can actually see things happening in the cell. So, when the cyclins were first discovered, they were discovered because the levels went up when DNA synthesis started. So they were called cyclin A. And then the next cyclin that appeared was cyclin B, cyclin D, cyclin E, and there is a cyclin C that is related to G0 phase of the cell cycle. So if you remember that for some people the cell cycle starts at S phase, you just remember it's A, B, C, D, E. The function of the cyclins is to activate enzymes called cyclin-dependent kinases, or Cdks. So each cyclin activates either an individual Cdk or a cluster of Cdks. So cyclin D activates Cdk4 and 6, cyclin E Cdk2, and so on. So at each stage of the cell cycle there's a different pattern of Cdks that are active. Now, the Cdks themselves remain fairly constantly expressed. They're just there inactive in the cell, waiting for their cyclin partner to be expressed. So the cyclins go up and down, and as the cyclins go up and down, the Cdk activity goes up and down, but the Cdk proteins themselves remain fairly constant in their expression levels. And one of the key events of Cdks that we're going to look at next is how cells get past the R point. So cyclin E activates Cdk, specifically Cdk2, and Cdk2 is going to phosphorylate the retinoblastoma protein that we're going to look at next. So that's the key event that drives cells through the R point. Now that Cdk2 activity, so we've got cyclin E produced in G1/S phase, that Cdk2 is going to be activated by cyclin E, and Cdk's job is to phosphorylate the retinoblastoma protein, which is controlling the R point. This is really important because retinoblastoma holds on to E2F transcription factors. It sequesters them. So whilst retinoblastoma is holding on to E2F transcription factors, those transcription factors can't switch on the S phase genes that are needed for S phase. If the Cdks phosphorylate retinoblastoma, the retinoblastoma will release E2Fs and they can go off to the nucleus and switch on the S phase genes. So this is a key step in S phase transition. Now this slide shows what's going on in the cell cycle with respect to retinoblastoma. So we've got our G1S, G2 and M phase, our R point is there. This little red line here represents the degree of phosphorylation of retinoblastoma. And it can be dephosphorylated at mitosis, and then it remains unphosphorylated, and then cyclin D Cdk4 tags on a few phosphates on there, not many, but at this point here cyclin E is expressed, Cdk2 is now active, and that hyperphosphorylates PRB, and that hyperphosphorylated state is retained all the way through the cell cycle until it's dephosphorylated at the next cell cycle. This means that the cell can't enter another S phase unless it's been through a period of G1 phase and responded to growth factors. Now everything that's driving G1 to S phase transition here by growth factors is being opposed by transforming growth factor beta. So TGF-beta downregulates the cyclins, and it will upregulate cyclin-dependent kinase inhibitors that we're going to hear about shortly. So we've got a balancing act between proliferative factors acting on this bit of the cell cycle to drive cyclin D and cyclin E, and TGF-beta acting to oppose all of those actions. This illustrates what's going on with retinoblastoma at different stages of the cell cycle. So here we've got retinoblastoma with a phosphate on there, but this is in G1 phase, so that might have been tagged on by CDK4. At this point it's still holding on to the E2F transcription factors. Remember, these are going to go to the nucleus, bind to promoter regions and switch on S phase genes. Once cyclin E is expressed, that activates CDK2, and that covers retinoblastoma with phosphorylations. Retinoblastoma changes its conformation, releases the E2F transcription factors, they go off to the nucleus, bind to promoter regions, and then whilst we're actually in S phase, they've all gone to the nucleus. We've got retinoblastoma lurking around. It's not bound to E2F transcription factors, it's just hyperphosphorylated. So it's not really doing anything here. But what we have got at this point is S phase gene expression. So whereas growth factors induce cyclins and cyclins activate CDKs, that alone might not be enough to kickstart the cell cycle, because the cyclin-dependent kinases have inhibitors which can stop the cell cycle even if growth factors are present. And one of the things that would stop the cell cycle would be TGF-beta. So if you might imagine a situation where you've got high levels of growth factors and high levels of TGF-beta, even though cyclin D is expressed, the cell cycle will not progress. And that's because of this group of Cdk inhibitory proteins, the INK4As. That means inhibitor of CDK4. CDK4 is associated with cyclin D. So these proteins, P15 through to P19, all inactivate the CDK4 cyclin D complexes. So even if cyclin D is present, CDK4 should be active, these proteins can come along and block that activity. And TGF-beta is certainly known to induce some of these. We've got another group of cyclin-dependent kinase inhibitory proteins. These are known as the CYPs. The ones you really need to know about is P21, but also P27, and these bind to the non-CDK4 and 6 group, so CDK1 and 2. These are going to block cyclin E and cyclin A induced events. So cyclin E at the G1S boundary, cyclin A in S phase. And these are really useful in stopping the cell cycle once DNA synthesis has already started. Now, the types of things that would stop the cell cycle then would be some sort of DNA damage. So the cell's trying to replicate DNA. DNA is damaged. You need to stop the cell cycle very quickly. Now, if you look forward to your P53 and apoptosis lecture, you'll find that P53 responds to DNA damage. It induces P21, and P21 blocks cyclin E CDK2 complexes and also cyclin A CDK complexes. So P53 can stop the cell cycle whilst DNA replication is occurring, and it does this by inducing P21. P27 does a similar role in other situations. Now this just summarises a series of events in proliferative and non-proliferative signalling. So in anti-proliferative signalling by TGF-beta, we get downregulation of cyclin D, so the cells are stuck in G1. We can get upregulation of P15 by TGF-beta, and this inhibits any CDK4 cyclin D that is already present, and that will stop the cells in G1 of the cell cycle. If you've got growth factors present, you'll get G1S transition by inducing cyclin D expression directly, so that's the end of that MAP kinase ERK pathway that I showed earlier. That will, by a system that I've not explained, induce cyclin E and CDK2, and that will result in phosphorylation of retinoblastoma. The final event is retinoblastoma releases its E2F transcription factors. They go to the nucleus and switch on your S phase genes, which are crucial for S phase. Okay, so this slide just illustrates all of the key points that are going on in cell cycle regulation. So all of these should make sense based on the lecture that we've just been through.